Hello, everybody, and welcome back to the SBGrid webinar series starting up in the fall. I hope that everybody in the Northern Hemisphere had a good summer break, or at least a okay summer break, and welcome back. This webinar is going to be recorded and it'll be up on the SBGrid YouTube channel. If you have any questions, you can send them to the hosts in the chat and we'll moderate at the end. Today we've got Daniel Meyer from UCSF and the home of the electrosome, and where they study mechanisms of ion channels in prokaryotic and eukaryotic systems. And Daniel, thank you for joining us, and take it away.
Okay, thanks very much. Okay, you see my screen?
Not quite yet, no.
Thanks, and thanks to everyone. So good morning from San Francisco. The air is not as clean as it looks behind me in the background, but it's better than you've been seeing on the news. It's improving. So thanks for joining today. And I'm going to talk about work from my lab, both about some work we've been doing on ion channels and talking about a class called K2P channels. And I'm going to have a little insert at the end talking about how to survive a toxic environment, which I think is a topic that is often on people's minds, especially these days. So voltage-gated ion channels comprise a very large family of signaling proteins. Actually, the third largest family of signaling proteins, just behind G-protein coupled receptors and kinases. And this is because we run on electricity, right? The electricity runs our crazy brain and heart and our nerves. And this is a very large family. Not everyone in this family is voltage gated, and we're going to talk about a class that makes a background current called K2P channels. This is an action potential here, and these channels are very important for setting the resting membrane potential of the cell. And so they control how excitable the cell can be. In humans, there's 15 of these channels. They have this unusual architecture. There's two pore-forming units within a single polypeptide, one here in orange, one in green. Two of these come together to make the channel pore, and so they're pseudo-tetrameric. There's 15 of these in humans. The founding member was one called TREC1. This is a channel that was known to be one of the first polymodal ion channels, which means that it responds to things like pressure, temperature, heat, so many different inputs. And what was known from early studies was that it's a target for analgesics, and knockout mice, which were made more than 20 years ago, have issues with pain. And so understanding TREC1 could provide some very direct connections to pain, as well as telling something about this entire K2P family. When we started our studies on this family, we really started with the question of how is it possible that one channel can listen to so many signals? And do all those signals converge on a single point to control the channel function, or do they all work in different ways? And we started by thinking about physical inputs like pressure and temperature, and also pH. And the way that we first addressed this question was using a yeast genetic screen, in which we did a forward genetic experiment in which we looked for gain of function mutations in TREC1 that would allow yeast that were deficient in potassium uptake to survive. And this gave us a surprise. It identified a mutant that told us about how the channel works. Oh, now you don't see my pointer, okay. And that was at the top of the ion channel near something called the selectivity filter, which is essentially the active site of these proteins. And the interesting thing about this mutation was it occurred in a site that in other potassium channels was a point of not where you would get a better channel, but where you actually get a dead channel. And so that already told us something about K2Ps, that they were different from other better studied members of the voltage-gated ion channel class. The other thing important that it told us was that this single mutant was able to blunt the responses to pH, temperature, and pressure. And so this let us answer that first question, which is, do all these signals converge on a single point? And the answer is yes. And that point turns out to be the selectivity filter, which was a bit unusual because most members of the voltage-gated ion channel family gate that is open and closed by using a barrier that's on the intracellular side. So our lab and Stephen Tucker and Thomas Braukowitz's lab in the UK and in Germany were able to show that first that these channels gate at the selectivity filter. So that's really something special about K2Ps. That's their primary gate. Now, the other thing that you should know about this selectivity filter, as mentioned, is like an enzyme active site. And it has something which is essentially like substrate-assisted catalysis. That is that the potassium ions that go through the filter, they contact the protein at this site, and they help organize the structure of the filter. And so you can see this really nicely with the functional experiment if you raise the external potassium concentration where you stabilize the filter structure. So here you can see an experiment where we try to close the channel with pH. In one case, we do this at low potassium concentrations. You see the channels are easy to close. And another, we do it at high potassium concentrations where the channels are difficult to close. Now, our gain of function mutant basically didn't care. It always behaved as if the filter was full of potassium ions. And so that was good evidence that the filter is really the gate that controls how these channels work. When we started this work, there were no structures, and Slav Bagriantsev, who had launched this work in my lab, started thinking about what this gain of function site might be next to, based on structures of other related potassium channels. And we reasoned that this part of M4, which is one of the transmembrane helices, might be close to this P1 helix from the first subunit. And so Slav started staring at sequences, and he realized that there was a site where some K2Ps had a large residue, but TREC1 has a small residue. And we thought they might be next to each other, where this first gain of function mutation was. And so he did the simple experiment of just making the small residue big and the big residue small. And that had the consequence of one, making the small residue big, stabilize the channel even more. So this was the first gain of function mutant here. In blue, you see the G to I mutation, right? These channels basically are very difficult to close. And if you do the converse experiment here in these task channels, you get the opposite phenotype. You get channels that now are much more easy to close. And so that helped us think that these guys are really close together. While we were trying to do some cross-linking experiments, we were saved from all that pain by the first two structures that came from Steve Log and Robin McKinnon's lab of other members of the K2P family that basically showed us that the two residues that we thought should be next to each other indeed were. And so what I want you to remember from this is two things. One is the beauty of forward genetic screens, right? Because we went into this with the faith that finding gain of function mutations would tell us something about the functional parts of the channel, that is the parts that move, that are important for how the channel works. And the second is that it identified this P1-M4 interface as being very important for channel function. I'm going to take a small detour into pharmacology, just tell you that like many ion channels that you see on that wheel that I showed you, K2Ps essentially have no usable pharmacology. This is a problem that still plagues the ion channel field in general. And we and others are interested in solving that problem and developing tools that let people probe channel function, both in model systems like transfected cells, but also in real physiological systems. And there's plenty of evidence that TREC1 and activators of TREC1 in particular could be useful for treating things like pain, stroke, anesthesia, and maybe even the bends that divers get. There's actually a paper implicating TREC1 in decompression sickness. And so Slav was able to use the yeast screen to identify what was the first selected activator of a K2P channel. This is ML6733, this dichloroacridine. This activates the TREC subfamily, so it's TREC1, TREC2, and TREC. We'll come back to those later. And we could show by mutational experiments and function that this compound worked directly on the ion channel filter. And you see it's much better than things people were using before to activate the channel that weren't specific, like chloroform, arachidonic acid. Both of these things, we think, work through affecting the tail. And so we basically have a connection where we think that the tail is a sensor for many inputs. It can affect M4's position, which eventually gets propagated to the filter to help stabilize the channel open state. We've not been able to get a co-crystal structure with our compound, but we were fortunate to get two structures with other activators that were discovered by Ono Pharmaceuticals. And in a really nice collaboration with them, Marco Lolikato in my lab solved the structure of TREC1 alone and with both of these compounds, ML335 and ML402. You see they're essentially equivalent activators of the channel. And we were able to see this L-shaped pocket that was behind the selectivity filter. And I will say this was in a way like a dream come true because we had imagined that this might be the position where ML6733 could work. And to actually see a compound that directly affected the filter was really, really beautiful day in the lab. This turned out to reveal something very interesting. This pocket is a cryptic site. And what I mean by that is it's a site that requires protein motion to open up. And so if you stared at the apostructure, which you're staring at here, and tried to guess where the compound would bind, you would never guess the site because it's a perfectly packed interface. And of course, what has to happen, and you see the movie, is that interface has to open up to make room for the compound to bind. So if you really, looked at this and tried to guess, you wouldn't even think that you could slide one thin mint in this interface. But in fact, you can. You can put this compound, this low molecular weight activator, in the site, and you see it's right behind the selectivity filter here. So cryptic sites have been well known in soluble proteins, but I think this is one of the first, if not the first, good example of a cryptic site found in an ion channel, and it certainly will not be the last one. It also highlights the importance of interfaces between the subunits as being important. You can think about this as an analogy. So back when we could still travel, I had the good fortune of giving a talk in India, and I was in Mumbai. This is a picture of me in the back of an auto rickshaw trying to get across the street so I can see a temple on the other side here. And of course, there's no path, but because the cars are in motion, when the gaps open up, my very talented auto rickshaw driver can slide us through the gaps and get us to the other side. Right? And that's the essence of the cryptic sites that we see in proteins. Because the proteins move, as they move between different states, they are likely to open up pockets that can be exploited to control their function. The structure that Marco solved also told us something interesting of other interesting things about channel pharmacology. TREC1 and many other K2Ps are modulated by lipids. And we were fortunate to identify a number of lipids in the structure, one of which you can see here, that basically occupies what is known to be, or what is the high candidate for the PIP2 binding site. So many ion channels are modulated by phosphate inositols, so PIP2 is being the major one. And you can see the tails here. And in fact, the neighbors here are all residues that are implicated from other work to be involved in coordinating the head group, which we didn't see here, but we've been able to see in a subsequent structure that I'll show you later. The other nice thing about this site is this is a site that's at a moving part of the channel. We know from multiple structures of TRAC, which is a relative of TREC1, from both Steve Brown and our lab, that this M4 helix moves. And exactly where that helix moves to open up this little window to the inner leaflet of the bilayer is where the PIP2 binds, we think. And so that's really sort of, again, a modulator that's sitting at a moving part of the channel. The thing about the Ono compounds that was amazing was that they were actually really selective. So the compound that Slav and I developed with Adam Renslow at UCSF activates everybody in the TREC family, so 3 channels, but not anybody else out here. The Ono compounds actually activate TREC1 and TREC2, but not TRAC. Now there's actually structures of all 3 of those channels, so as a good structural biologist, you should be able to explain where the selectivity comes from. The binding sites, or the residues that line that pocket, which we call the K2 modulator pocket, are identical among these 3 channels, with the exception of one residue, which is a lysine in TREC1 and TREC2 and a glutamine in TRAC. Now we could see in the structure that this lysine made a cation pi interaction with the upper ring of the activator compounds. And so we did, the simple experiment. We said, what happens if you flip the lysine and glutamine between TREC1 and TRAC? And what happens is that TREC1 becomes insensitive to the compound and TRAC becomes sensitive to the compound. And so that's really a beautiful example of one residue and a cation pi interaction, right, which is generally something that's not worth a lot of energy, or maybe a KT's worth of energy, and half a kcal, or maybe 1 kcal, and that's enough to control the selectivity of this protein, of the compounds. We think that this modulator pocket will be a general site that can be targeted in other K2Ps, and what I just want to point out here is that this is just looking at the diversity of this site. So everywhere you see is blue is where residues are identical to TREC1. Everywhere that you see red are residues that are different. And I've opened up the site like the pages of a book, so you can see both sides of the site. And what you can see is that it's really quite diverse. And so we're optimistic that this site will be targetable by other small molecules. We wanted to show that this compound really directly activates the channel. And to do that, we did some electrophysiology. So this is a patch clamp experiment where we remove a patch of membrane that's expressing the channel. And what you see about TREC1 is it does something called rectification. So for those of you who aren't electrophysiologists, all that means is that it conducts more current in one direction than the other direction. So you see there's no inward current, there's lots of outward current. Now, Thomas Barkowitz and Stephen Tucker showed very nicely that almost everything that activates TREC1 and other K2Ps turns these channels from being outward rectifiers into leak channels. And so that current that is bent like this becomes very straight in this kind of experiment. And so we did that with our compound, and you can see that basically ML335 directly activates the gate, as it should. It's sitting right behind the selectivity filter. And the gain of function mutant that I described earlier, that was very stable gain of function mutant, does exactly the same thing. So what's the lesson here? The lesson here is again about this P1-M4 interface. It basically says if you put extra atoms in this interface, either by delivering them through a compound or by mutation, you can activate the channel by stabilizing the filter. And so this brought us to a model of how we think about how K2Ps gate. So these channels, they're not digital switches, right? They don't go on and off. They basically go from being quiet to being more active. So it's sort of a rheostat kind of change where you can turn the activity up and down sort of in a linear fashion. We think that at rest, the channels equilibrate between a mobile state where the filter is mobile and there are not enough ions in the selectivity filter, and an activated state where the filter is stabilized, this is a more rigid state, and the ions can go through. And the compounds that I just described to you basically act like little wedges that act like putting the wedge under the door, right, to stop the motion. And so I'm just going to make a brief summary of some points I want you to remember here. One is the importance of stabilizing interface and ion channels. Now, this, of course, is not a new concept. We've known for many decades that the acetylcholine receptor, right, one of the model ion channels, where does the ligand bind that opens the channel? It binds at an interface between the subunits. Right? And you can think these are the points in a protein where normal mode kind of motions might be very easy and might be things that could be very easily exploited. We think that this kind of model provides an explanation for how it's possible to have many things activate the channel, because no matter how the input comes to the gate, all it has to do is stabilize the structure. Right? And so there may be many ways to do that. It turns out that M4 up and M4 down can both activate the channel, and we think this provides a solution to what appears on the face to be a paradox about protein motion and gating in these channels. We wonder if there's a natural effector. Obviously, this site was not put there for these ONO compounds to bind. We don't know if something binds there, maybe a lipid, maybe a protein, but we're very interested in finding this out. And as I mentioned, the subunit interface are really important. Now I'm going to talk a little bit more about pharmacology. So there's a compound called ruthenium red, which is shown here, is a triruthenium oxo-bridge compound. It's supercharged, it's +6, and it has a more than 50-year legacy as an ion channel inhibitor, inhibiting many flavors of ion channels, that you can see here, including K2Ps. And we were interested in how this works. There were no structures of ruthenium red bound to anything when we started. And we thought we could take advantage of the fact that it works on K2Ps. And what was known from really nice functional studies from Peter Igneti's lab in Budapest is that this extracellular structure, which is called the cap, which is a unique structure that's only found in K2P channels, that the determinant for ruthenium red binding is an acidic residue that sits at the bottom of this cap. And the two K2Ps that are sensitive to ruthenium red, TREC2 and TASC, both have an acidic residue here. You can make TREC1 ruthenium red sensitive by making an I to D mutation at this site. That's what my student Leanne did here. You can see the channels can be blocked with ruthenium red. The charge matters. Glutamate still works. It's not as good as aspartate. And Leanne was able to get crystal structures. And of course, the nice thing about working with ruthenium red is you don't have to guess if you have the complex. You see this beautiful magenta crystal here that told us that the ruthenium was in fact inside the crystal. We could visualize the site. It binds under the cap at a site that we've termed the keystone inhibitor site, because what the cap does is it makes an archway above the selectivity filter, right? And that's the keystone of the archway where both of these residues that coordinate the ruthenium red bind. And this is just a morph between the apo and bound state. And the only thing I want you to highlight here, here's these two aspartic acids, here's the selectivity filter. What you want to see is that there's these holes, right? These are the pathways that the ions would come out, and ruthenium red comes in and blocks essentially the exit pathway for the ions. You can see that here. And so we call this a finger in the dam type of mechanism, right? So normally what happens with these channels, the potassium is flowing out, it comes to the cap and it goes left or it goes right through these two exit pathways. And ruthenium and other compounds like Ru360, which is also a ruthenium compound, is able to come in and block the channel, just like the story of the boy who put his finger in the dam to stop the floods in Holland. Okay. So what we've learned from all of these studies, and some studies from the group in Oxford from Stephen Tucker and Liz Carpenter, is that these channels, even though they're small, so these are 70 kilodalton complexes, are rich with modulatory sites. So I've told you, we'll start from the outside. So there's a keystone inhibitor site, which is an outside site that can be blocked by charged polyruthenium compounds. There's this K2P modulator pocket where these activators discovered, characterized by us and owner pharmaceuticals bind. There's a lipid modulatory site where we think PIP2 binds on the inside. And then there's a site here called the fenestration site. So when that M4 helix moves up and down, it makes a gap that's just under the selectivity filter, where there's a window that's basically looking out to the middle of the bilayer. And the Oxford group has nicely shown that two different types of compounds bind there. Norfloxetine, which is an inhibitor, but also it's a likely site for this activator, BL1249. And that's really curious because you have a site here where both, we think, occupation of that site by two different compounds can have two opposite outcomes, one of which blocks the channel and one of which activates the channel. Now, how that actually works, we still don't completely understand, but I think it's really fascinating sort of puzzle to still be solved. But what this shows you is that for a channel that had no pharmacology, right, after a number of years of work, you see this is very rich, and we think this is going to be true for many members of the family. So, you know, I've been pulling your leg a bit here, right? I've been talking about the filter, which is also called the C-type gate. I've been telling you it's important that the potassium ions organize it and all these things. I haven't shown you a single structure where the filter changes. And then that's, in fact, because there aren't any, or there weren't any. So this is a superposition of all the K2P structures that have been solved up to about a year ago. They have different states, M4 is up, M4 is down. They're different channels. You see that all those filters look really the same, right? In fact, you can zoom in and look here and you can see that. So look, what's going on, right? Am I selling you a story that's not true? And the answer is no, right? And so why is this, why are these structures that way? Well, if you look at how they've all been solved, they've all been solved using conditions that are very high potassium concentration conditions. So 150 to 200 millimolar K. You might expect that that provides enough ions to stabilize the filter. And so we wanted to ask whether we could get some structures with lower potassium concentrations and see whether we could watch this gate move and see what the consequence of that is. And so Marco Olicato was able to do that. He solved structures of TREC1 in 7 different conditions, and actually it's 14 structures because it's with the ML335 activator and without. And I'm not gonna show you all 14 structures, but I'm gonna show you the summary of this. So when the activator is present, it's a very boring answer because the high potassium and the low potassium structures look identical, as you might expect if that activator compound is meant to stabilize the selectivity filter. It's a completely different story if you solve those structures without the activator present. The 200 millimolar structure in the absence of the activator looks just like these on the bottom when the activator is present. We see everything. The filter is very well organized. But as the potassium concentrations go down, we see the filter start to unravel and unfold. And you can see that at its most extreme here in the 0 millimolar K, where the filter is here, and then this loop, this SF2M4 loop, essentially disappears. We can see in these structures that the ions also are emptied from the selectivity filter site. And we were able to do a really fun experiment. I had the good fortune of meeting Armin Wagner at a meeting about 1.5 years ago. And Armin at Diamond in the UK has developed a long wavelength X-ray beamline that allows you to look at biologically relevant ions. So we could measure with Armin and Ramona the anomalous diffraction from the potassium ions. So we don't have to put in a proxy for the filter. We actually see what we want to see, which are the potassium ions in the filter. So what I'm showing you here are anomalous difference maps at 200 millimolar K and then at 1 millimolar K. And here you see, right, field filter. What happens in the low K structure? You see that the top two ions are gone. Right? There are ions at these two lower positions, but not at the upper positions. And a summary of that is shown here. So this just shows you if we look at ion occupancy as a function of potassium concentration at the 4 sites that are in the filter, around 50 millimolar potassium, there's a transition between being full and only having 4 ions and having only 2 ions. And you can see that in these morphs between the 200 millimolar and 0 millimolar structure, the consequences to the 2 filters. So I mentioned before that there's 2 filter units in 1 polypeptide chain. And what that means is that these channels are inherently asymmetric because the sequence of filter 1 is not the same as filter 2. And that's going to become very important now for this part of the talk. And you see that filter 1 basically pinches. You can see these asparagines coming together here, and filter 2 unravels. Now, we wanted to look at this in more detail and to try to get a feel for the dynamics. And we've been able to team up with our colleagues on the floor, on our floor in the CVRI building at UCSF, and that's Andrew Natali, who's a joint student with me and Michael Grabe, to do some molecular dynamics simulations. So Andrew did lots of simulations. I'm going to just show you some movies. This is the first filter here. This is the second filter. And what you can see is that when we go many microseconds, in the absence of the compound, this is the orange, the filter tends to enter these states that are non-conducting and basically often where it never returns. Whereas when the compound is present, we have an increase in the current and an increase in conductive events. And if Andrew analyzes the structures, you see something that exactly matches what we see in the crystal structures, which is that selectivity filter 1 has some disorganization that happens that results in a pinching at the top of the filter, whereas selectivity filter 2 basically unravels. And so we have 2 mechanisms operating in this filter. Now, I've been telling you that compound activates the filter. The simulation suggests that it stabilized the activated state. You would really like to see it. And Faisal Abdurrahman Ali in my lab, a super talented postdoc, has been able recently to get single channel recordings of TREC1 with and without the activated compound. And it's obvious here what you see. You see that without the compound, TREC1 rarely opens. When the compound's present, we have many, many openings of the channel. So there's no change in the single channel current amplitude, but the NPO goes up. Again, this is the same story. Those wedge compounds come in and they stabilize the filter by reducing its degrees of freedom. Right? So Andrew noticed something else as we were going through the simulations, and that is that if you look at K2P, so almost every potassium channel, right, which has this shared architecture in the pore, between the selectivity filter and the outer membrane helix that's linked to the selectivity filter, there's a linker that's almost always 6 residues long. And the first domain of K2Ps has a 6 residue linker, but the second one has a much longer linker that varies between about 12 and 14 residues. You can see that structure here. And what Andrew noticed in the simulations is that there was a network that was helping to stabilize this loop that's centered on this conserved glutamate, which is found in all the K2Ps. At this position, and it's a hydrogen bond network between this glutamate, this tyrosine, and the backbone. And so we asked the simple question, what happens? We could see that network fall apart in the simulations, and when that falls apart, this loop actually starts to have much more freedom of motion. And so what happens if we disrupt it? So if you disrupt this network here, you basically blunt the response to physical inputs. So temperature, the response goes away. Same with pressure. Right? And so this, again, is the key part of controlling the function of this channel. You can see that black residue here, or this black molecule here, that's the activator. And so that loop basically is sandwiched between the P1M4 interface and the other side of the P1M4 interface. And so you can stabilize it with interactions from the channel or interactions from the small molecule. And so we have 1 channel, 2 mechanisms. So what I want to tell you for the channel aficionados who are listening, they know that arguments about potassium channels inactivated at the filter have been going for a long time. And there are essentially many models, two extreme ones. One is that there's some pinching motion that happens, and the other is that there's some kind of dilation. And what we've managed to see here is that this channel actually does it both ways, right? So the first filter pinches, the second one dilates, right? And we think this is really important for giving K2P's interesting properties. It may explain how you get channels. So K2Ps are often homodimers, but they can be heterodimers, and the heterodimers always have different properties than the homodimers. And we think that some of those properties come from this asymmetry in the filter. So this asymmetric gating is really the key thing. So I'm just going to sort of summarize this work here on the filter and the channel biophysics to say, you know, if you look at everybody in this family, there are now structures of essentially representatives from every branch of this family. And you look at the core structure that makes the filter, right? Or that makes the pore from a single subunit. They're basically all the same, right? It's like if you don't work on kinases, right? One kinase looks just like all the other kinases, right? Same thing here. If you look at one pore, all these pores essentially look the same. This is just the superposition of two bacterial sodium channel pores with the KCSA potassium channel. You see the essential elements are this pore lining helix. This helix is faces the lipid, and the pore helix here that helps support the selectivity filter. Now that gives you two kind of gates that nature plays with. One is this inner gate where the channel can close at the bottom and block ions. That's a gate that's not used in K2Ps, but the other is the selectivity filter gate. And we now see that selectivity filter gating is used in many members of this voltage-gated ion channel family. It was known in shaker type channels from studies a long time ago, from Rick Aldrich and others. We and Stephen Tucker's and Thomas Barkowitz's group have shown that this is really the key gate for K2Ps. There's nice examples from the trip channel family that shows that the filter acts as a gate. And my lab showed recently that the filter actually acts as a gate in voltage-gated calcium channels. And so it's not a surprise, right? If you have this common architecture, there are ways to use the same themes over and over again to different degrees. And one challenge for the field now is to understand how you can make this different, how we can exploit it, and how these motions are coupled to the motions that activate the channel and to compounds or toxins or proteins that modulate the function. Okay, so in the last 10 minutes or so of the talk, I'm going to switch topics to something that is a new area of research from my lab that's really exciting, and that's about how to survive in a toxic environment, which I think everybody has ideas about what a toxic environment is, and I'm sure you've all experienced them to one degree or another. And you might like to know how nature kind of deals with this problem. And so if you work on ion channels, right, there are lots of toxins that target ion channels, and there are lots of poisonous organisms. And we got interested in this general group of toxins that target voltage-gated sodium channels. And I'm showing you three here. There's two guanidinium toxins, tetrodotoxin, saxotoxin, and this other toxin here, batrachotoxin, which is found in poison dart frogs and the hooded pitahui, which is a poisonous bird from Papua New Guinea. All these things target sodium channels. These organisms carry very high quantities of these toxins, and the question is, why don't they poison themselves? Right? Humans, of course, have learned how to exploit these toxins. This batrachotoxin is famous because South American natives use it to hunt. That's why they get it from the poison dart frog from their skin. Of course, humans have learned to titrate tetrodotoxin so it gives them some pleasure. This is a fugu dish of fugu sashimi that didn't kill me in Japan. And I can say it was very nice, and it generated some numbing feeling in my trigeminal ganglia, which would definitely help by sake, but very nice. Right? And then there's this other one here, saxotoxin, which is concentrated in clams, and we're going to talk about. Saxotoxin was quite famous in the 60s because this guy here, who old people in the audience may know, is Gary Powers, who was shot down over the Soviet Union in the 60s in a spy plane. And he was carrying this coin, and inside the coin was a pin, and on that pin was an amount of saxotoxin that he was supposed to use for suicide, to not give up the government secrets. That didn't happen. He ended up coming back in a prisoner exchange and was actually reviled when he came back to the US for not using the pin. I can believe that. So what is saxotoxin? So saxotoxin is a class of guanidinium toxins. It's made by red tide, which many of you may have heard of. This is a outbreak of microorganisms generally in the ocean, but it can be in fresh water. And it's super toxic, right? So the amount that you need to kill somebody by ingestion is 5.7 micrograms per kilogram. Just to put this in perspective, tetrodotoxin is maybe 80 times less lethal. Cyanide is thousands of times less lethal. Because saxotoxin is so lethal, it's a class 1 chemical, schedule 1 chemical weapon. The CIA had actually stockpiled it, as had other countries, until 1967, when there was a treaty signed that was a ban on these. I put until in quotes because the CIA was supposed to destroy it, but they didn't. It was a good thing they did, because the amount that they had collected has been used for research. It causes something called paralytic shellfish poisoning, which is a hazard that I'll discuss, and is one of the other things, aside from fires, that we have to deal with in California. So if you wanted to know how much tetrodotoxin it takes to get rid of me, there it is. It's not even half a milligram. And as I said, this paralytic shellfish poisoning is a problem. It was first identified on the west coast of North America. We have yearly bans on collecting shellfish, because if you ingest shellfish that have the toxin, you can die. And actually, all the shellfish that's collected commercially for human consumption needs to be tested for this. And one of the leading assays is a mouse lethality assay. If you can believe it, what the fishermen have to do is make an extract of the shellfish that they collect. The extract is injected in the mice, and then you just look to see if the mice die. Right, so like many things that happen with climate change, paralytic shellfish poisoning outbreaking is increasing as the oceans warm. This graph just shows you the difference in incidence of paralytic shellfish poisoning between the 1970s and 5 years ago. And you can see it's a big problem, not just on the west coast of the US, but Southeast Asia, where lots of our seafood is caught, in Europe and other places. So it's a growing problem. So how do you survive this toxin? How did the clams survive? So Bill Catterall's lab discovered this a number of years ago, and it's a beautiful answer, right? It's one that you might think shows up, which is mutate the target. The toxin binds a sodium channel. Maybe you can make a sodium channel that's resistant to the toxin. And they discovered one point mutant in the sodium channel that reduces the sensitivity to the toxin by 10 to the third. That's pretty amazing. This toxin blocks the pore of the channel. It blocks the active side of the channel. And this mutation, surprisingly, doesn't compromise function, or doesn't compromise function enough, but allows the clams to survive. Unfortunately, that's also why they can collect the toxin and then maybe poison us. So this story is actually a nice story because it has roots in UCSF. So this poison, saxotoxin, was first discovered by Herman Sommer and Chauncey Leake working at UCSF in the 1930s and collecting poisoned shellfish from the shores here around San Francisco Bay at the right times of year to isolate the toxin and characterize it. And what they discovered was that, they were injecting the toxin into various critters that they could find. What they discovered was that frogs happen to be really resistant, right? And this is written here. It's basically, you can't kill the frog with the toxin, right? So why is that? We know that the frog sodium channels are sensitive to the toxin. So maybe there's some other mechanisms besides mutating the target to allow these guys to survive. There was a protein that was discovered in the 80s that's called saxophiline, which is a high affinity saxotoxin binding protein, nanomolar affinity, found in the cytosol and the myocardium of frogs. It was characterized in the 90s by Ed Machowski's lab. And my lab got interested in this to try to see whether we could understand what the principle was for how this protein recognized the toxin, how it might protect the frogs from being poisoned, and whether we might be able to turn it into either a sensor or maybe a way to treat people who have paralytic shellfish poisoning. Because right now, the only thing that they can do if you get poisoned with this toxin and figure it out in time is put you on a respirator. And then either your liver gets rid of the toxin or the toxin gets rid of you, right? And so we thought there has to be a better way. So we were able to make saxophilin. It's a secreted protein. We tag it with GFP to make it easy to purify. We can get enough, you can see it from the other side of the room. It's beautiful protein purification. And Tianrei Yan in my lab was able to solve the structure of this alone and bound to saxotoxin. And what you can see, this is the structure of the protein here. It's a protein that's related to transferrin. This was known from the sequence, right? Transferrin is an iron transport protein. It's different from transferrin in that it has some protease inhibitor domains stuffed into the middle. These are thyroglobulin domains. We don't know why they're there, but they're there and they're probably used for something important. This is where the saxotoxin binds. It binds on one of the lobes of the transferrin-like fold. This protein can't bind iron, right? So the residues that are normally conserved that are in these two clefts here where transferrin would bind irons have been mutated away. It doesn't bind iron, that's been shown. Instead, there's a new site that's used to bind saxotoxin. And that's curious because when the protein was first characterized, the first hypothesis was maybe the iron site was mutated into a site that could bind saxotoxin. But that turns out not to be the case. We have this other site that's on the C lobe. There's nothing interesting that happens between the apo and bound structure. You can see it here. It's essentially a pre-organized site. There's one residue that moves, and we think maybe this explains in part why the affinity is so high, because it's really pre-organized. It binds a very rigid, it's a rigid site that binds a rigid toxin. And you can see that the principal interactions are clamping those guanidinium groups with acidic residues and a cation pi interaction that comes up and makes an interaction with the bottom of the saxotoxin framework. It turns out that this is the same framework, or this is the same strategy that voltage-gated ion channels use to recognize the toxin. Right? And so this is a superposition of the structure from Ning Yan's lab of saxotoxin bound to a sodium channel with the residues that coordinate the saxotoxin there, with the ones that coordinate saxotoxin and saxophiline. And you can see that this is amazing convergent, right? Even the channel also uses this cation pi interaction, but instead of coordinating the ring on the right, it coordinates the ring on the left, right? But it's basically the same principle. A rigid site, right? Ionic interactions to clamp the guanidinium groups and cation pi. And that's amazing because, you know, one of these is a soluble protein and one of them is a membrane protein. So I think it's a beautiful example of convergent rules for molecular recognition. And I think you can tell us a lot about how we can improve or design other proteins that might help us detect saxotoxin. Okay, so this structure brought up one other question that I want to finish with. And I'd really be curious to hear input because there's a question like, how do proteins evolve new small molecule binding sites? Where do they come from? Like, can we learn something about that? And so we just thought we could look to see what is this site where saxotoxin binds and saxophiline look like in transferrin. And what you can see is that there's not a binding site there per se, but there is what I'll call a molecular divot, right? Where there's a crevice and you can see that maybe with some rearrangement, this is just that crevice shown in the same lobe from transferrin from two different structures. And maybe something binds there. And we wonder whether transferrin uses that molecular divot to carry other compounds. And that maybe was the substrate for the development of the high affinity saxotoxin binding site, right? Because otherwise it's improbable. You can't go from a binding site that's not there to a binding site that binds in at nanomolar affinity in one step. And to my surprise, I had expected that there would be a long literature on where small molecule sites evolve from in proteins, and in fact, there's not. And so I think this kind of example provides a good example to start digging into this really fundamental question about protein recognition. So how do you survive a toxic environment? Well, one is you can mutate the target, but it actually might be more efficient to develop a toxin sponge because this allows you to interact with many toxins. So what we learned from studying saxophiline so far is these convergent rules for saxotoxin recognition that involve a rigid site and cation pi interaction in acidic groups. We know that there are other frogs that have this protein. We identified one in a Himalayan frog, and we're quite sure that there are other saxophilins out there. We wonder whether there are other toxins used. Lauren O'Connell's group at Stanford has studying poison frogs independently has discovered the importance of saxophilin in responses to feeding frogs with toxic compounds. And so we wonder if this is, again, part of a general response to toxin and surviving toxic insults. We're curious about whether we can turn saxophilin into better assays than this really brutal mouse lethality assay for looking for saxophilin, for looking for saxotoxin, and whether we can use it as a treatment for paralytic shellfish poisoning. And we also just wonder, like, what happens once the frog scoops up the saxotoxin? Where does it go? Is this related to the fact that frogs are able to store many poisons in their skin? And these are all questions that now occupy a lot of our thoughts in the lab, and that we have a lot of enthusiasm for chasing. So the most important slide here, of course, is to thank the people who did this work. So the K2P work was started by a very talented postdoc who's now a professor at Yale, who studies mechanoreception in ducks. That's Slav Bagriantsev. Marco Lolicato is the person who launched all the structural studies. Christina and Fayal did much of the electrophysiology I showed you. Leanne solved the structure of the ruthenium red compound complex and did lots of the characterization there. Tianrei is the person who developed the saxophiline expression system and solved the structure, and got that launched in my lab. We've had a great collaboration with the Small Molecule Discovery Center and Michelle Arkin, Adam Renslow, and Cliff Bryant at UCSF for our small molecule work. We've had this great collaboration with Michael Grabe and Andrew on the molecular dynamics, and Armin and Ramona to do the long wavelength anomalous scattering that I described. And then Justin Dubois and his lab at Stanford. Justin is the one person in the world who knows how to make saxotoxin and saxotoxin derivatives. And it's a great collaboration. And we've been funded by the American taxpayer. So for those of you who pay your taxes, thank you. And also through this collaboration with Owner Pharmaceuticals. And I'm going to stop there. And I'm going to say, when it's not smoky out here, this is what it looks like. We work here when we're not working from home. And if you want to join us and fight climate change one protein at a time, get in touch. So thanks very much, and I'll be happy to answer any questions.
Thanks, Daniel. That was a really cool talk. Lots of interesting areas, covering things in different places. So we've got a couple questions. One that was maybe you alluded to towards the end, but for the poison arrow frogs, do you think that they use the sponge approach or the resistant receptor approach?
That's a very good question. Yeah, we're not sure. We're investigating that right now, and I think there's going to be a very interesting answer, and I think I'll leave it at that.
That's how a lot of the interesting research questions are. You know, what's the answer? We don't know. Let's find out.
Yeah.
So we another question from Suravoy, and I apologize if I'm mispronouncing your name there. Is the size of the opening of the cryptic site, is that modulated by the molecular size of the small molecules?
It doesn't seem to be. No, it's that site is basically the site of those diaromatic compounds. It's not much bigger, and they basically, the lower ring basically fills the base of the pocket, and in the ML335, the sulfonamide actually pokes out from the top. So that's essentially the entire size of that cavity. Yeah. So it's not much bigger, and we know that if we make bigger things, they stick out. Yeah. Yeah.
I guess kind of also related to the cryptic sites, is there in the apo structures, is there any indication from the B factors that this might be something that could?
No. And in fact, if you look, there's a figure in the paper, but if you look, I mean, those B factors go down when the compound binds, but they don't start off being very high. And if you use programs that are meant to identify cryptic sites, it doesn't. It's not findable. Yeah. Again, a good reason to actually do the experiment.
Yeah, exactly. Yeah. And you've probably looked at, like trying to do normal mode analysis on the structures and such, and I would imagine you'd get a similar thing there.
We haven't done that so much. We focused first on this molecular dynamic simulation to look at the things in the filter, but I think it'd be a good idea to now ask, like, how much other motion on sort of a larger scale is there in that interface. Yeah. We've also been interested in doing docking screens to see if we can identify something that binds there, but so far the best things that bind are the things that you see from the structures. Yeah.
That would probably be pretty tricky. You'd have to kind of mix dynamics and search at the same time for the docking. So another question from Ming Zhao is there any hint of C-type inactivation for the K2P?
You know, I think so. I guess the answer is yes. We're calling it C-type activation, but the converse is the inactivation. So the C-type inactivated state is the low occupancy state that you see, and we see that in the simulations, and you can see it in the single channel records, and that's consistent with what we see in the. We don't see, you know, there are not other kind of rearrangements like have been described by Ming and others in KCSA and these types of changes. But it seems sufficient to stop the ions to basically pinch the top of the filter, lose those top two binding sites, and unwind the S2M4 loop. But you know, what's interesting is that for the potassium channel aficionados, that glutamate that we identified in M3 corresponds exactly to the same glutamate that's been identified in voltage-gated channels that controls C-type inactivation. So it's clearly some kind of very conserved mechanism. The twist here is that it's supporting a long loop instead of a short loop. So.
Another question, is there potential for use in the compounds for pharmaceutical purposes? And if there is, would it make more sense to activate or inactivate?
So it depends on that. So it depends on the application. So for pain, activating K2Ps is a very good idea because they're in the same neurons that TRPV1 are in, right? So the target of capsaicin. And activating the K2Ps has the opposite effect. So instead of causing pain, it actually stops pain. And we think that maybe having a compound that works on a channel where you can tune the activity sort of up and down continuously could be more effective than going after a channel where you have to hit a digital switch. So we've been interested, and we have a number of collaborators we're working with to try to get to see whether these compounds work in various in vivo systems.
Cool. And I guess a follow-up for me, not from the original questioner, but you'd mentioned the bends as being involved in the system. Yeah. Is that just the pain from the pressure?
It's due to, yes, it's involved. So this channel is involved in ischemic recovery that happens. And so when you have the block, when you have ischemia happen on the rebound, this channel is very important. And so you could imagine that from what's known out there from the experiments that have been done is that activating the channel. So what's known is that the knockout mice that don't have TREC1 suffer the bends much more than normal mice. And so that implies that if you could activate the channel, you might protect divers who, you know, come back, which I think also would be a neat application, although we don't have a way to give mice the bends. That was the French Navy who did that experiment. But I think it'd be a fun application. Yeah.
It's definitely, you know, not the first thing that I would have thought about when you think about voltage channels.
Yeah, but for the same reason, it could also be important for stroke, right? Other places where ischemia and ischemic damage can be mitigated.
So another question: could you comment on the role of the selectivity filter gating by temperature in the K2Ps and the TRPs?
I think that the examples, so the selectivity filter is important in some trip channels for gating. I think that there's quite a bit of controversy around how important it is in different members, and I just want to say I think it's important. It may be related to what we see going on here, and I think we need some more things done to figure that out.
Okay. I guess one other kind of speculative or interpretive question is, do you have any thoughts on how the tail domains might modulate might propagate their changes to the gating areas?
So what we know is that if you uncouple the tail from the pore, so Slav did some of these experiments with some glycines, you can get rid of the temperature modulation of the channel. And so, you know, the general idea is it's kind of like the shin bones connected to the knee bones connected to the thigh bone, right?
Yeah.
That the
Yeah.
tails move M4, and because M4 repositions, it's able to change the dynamics of the filter. Exactly how that works, we still need to figure out. And, you know, I think a big step there will be seeing these channels imaged in lipid bilayers, because we know that the bilayer is important for how they work, and certainly we're missing things from these crystal structures because we're trapping states in the absence of the lipid bilayer, and especially for that tail, which is essentially an extended helix that you could imagine, and it's a empathic helix, and so it should interact with the bilayer in interesting ways. Yeah.
It's just not a question, but as a crystallographer, I found it pretty cool that with the anomalous scattering experiments you guys did in the channel.
They were great. I think that's also the example. I mean, Zoom things are great, and this is a great way to interact with people, but there's no substitute for being in person. And that Armin and I happened to be at a meeting together at LBNL, and I heard his talk, and I just went to the coffee break, and we started chatting, and it was, you know, it was that kind of, those kind of interactions are really important in science, and I think all of us are really enthusiastic to get back to them at some point in the future.
Yeah. So I think we have one more question from HHV, which I'm just going to read this directly because I'm not sure if I'm going to get the mechanism right. But if how is the pinching dilating of the filter involved in the directionality of the ion transfer?
Oh, so the ion's set up by the gradient. So there's more potassium inside than there's outside of cells, and so in a regular cell that's not being, where its membrane potential is not being controlled by a patch clamp instrument, the ions only go one. The potassium ions go one way; they go out. Right?
Okay.
Because once you get to EK, they stop, and to get below EK is very difficult unless you do things like plant cells do with proton gradients and things that can make the ions go in. But basically, the direction set by the ionic gradients. Yeah.
Cool. Thank you. So I think that's all the questions we've got, and you know, we finished exactly on time, which I don't know if I did not plan that. I think that's just we ran out of questions. But thank you again for presenting, and thanks everybody for coming. But again, that was a really interesting talk, and I learned lots of cool things about ion channels that I hadn't known before.
Thanks.
And a little bit about living in a toxic environment.
Thanks.